All right, this is how the whole thing unfolded. This goes back to March 4th, 2015. I contacted this guy at Yale who was, I thought he was the big shot in charge of this stuff, but he says, no. Well, here's let me just read it to you. I said, hello, and this is to Derek Briggs. Hello, my name is Roger Spur. Professor Berger requested I contact you regarding mud fossilization. I thought I was supposed to speak with him, but he said, no, Derek Briggs, you have to go through him. I said, okay. I said, I'm working with wet platy silicate mud rocks, which are thought not to contain fossils. This is in Connecticut. I'm 15 minutes away from Yale. They don't contain bones like the dry desert sands, but they contain actual muddy, rocky copies of the creatures and some with possible DNA, which I ended up doing because they wouldn't speak to me. The encapsulation was quick, cold, compact, anaerobic, platy, polar silicates. That's the key. With continuing wet conditions resulting in microcurrent fossilizations, nucleophilic invasion. Process literally electrical replacement, nucleophilic invasion of the original matter, because Ca2 plus is so reactive in a solution. And then I just detailed the whole thing to him. And I said, I have a YouTube that describes the process, examples of the perfect fossilization. I appreciate it for an opinion. And I think he did send me back something. He says, oh, so there's just rocks. And that was basically it. And from then on, it was just a fight. And I have 20, 30 emails between me and him. And I'm going to give you his email and his phone number. As a matter of fact, it's right here. Derek Briggs. That's him. And this is his phone number. And it's derek.briggs@yale.edu. I think you should ask him why he won't correspond with me. And as far as I can see, he is saying precisely what I told him. And that was a year later. And who funded that study was NASA, because I contacted them too. I said, Yale won't talk to me, but you guys got the same stuff everywhere on Earth. And they're also on the moon and on Mars, because they had just had a curiosity up there. There's no question, 100% certainty, 100%, that Mars has exactly the same biology as here, only Mars was killed, destroyed, because of an impact from Venus. And you can see the Marianas Trench is huge. It's wider than the United States. And Venus has this big welding arc, which sucked in all the magnetic field, all the electrical energy of Mars. We got to look back at our reality, my friends. But this is the kind of problem we're having here, is with this kind of thing right here. I'm going to show you what he put out, his paper, a year after mine. All right, if you're going to have to read this paper, it's exactly what I said was from heavy silicates, which came up from the ocean floor. It's called a siliceous ooze. And it's heavy. It's 2.5 times heavier than water, I believe. And it goes to the bottom and it creates an ooze. When we almost got hit by Venus, or did, all of that stuff came up in the ocean, coated the whole world. And they say this is where it happened, in this exact particular layer. It says this Ediacara biota, which means the preserved creatures, Earth's earliest fossilized ecosystem, which means there was nothing before. There was no. These are the first things that ever showed up on Earth, is what they're saying. They were complex, macroscopic, multicellular organisms. A slime, basically, is what they say. It occurs in this terminal Ediacara strata worldwide. It's in this one layer because the whole world was sloshed. And it all settled down, and it had a red bed, which is all the boiled flesh, because it got hot, very, very hot. Originally, I thought it was cold. I didn't take into account the impact of Venus and heating the atmosphere and so forth, but it would have boiled the oceans. And now they say, yes, the oceans did boil at some point in time. But this is the key. It killed everything worldwide, basically, just as it says in the ancient text. And they say it consists of exceptionally preserved soft body forms. Many of the fossils are anatomistically preserved in casts, yeah, like this. They were in that mud. And then now the mud's run off. There's still a ton of rocks that are in mud that when they erode out, they're going to be mud fossils. There's just nothing but it. And some of them are like this. It's just meat. It depends on where it was when it got entombed. I am up at the top. As eventually all this stuff ran off when I believe some big lake they claim, because this was a basin originally in the area I am, which means it's a boiling pot. And all these things just boiled and can boil and boil and boil. I'm sure, I don't know how long. But at one time, eventually, a dam broke, they claim, and voided this whole area, and at that time pulled all the mud away. And what was ever under that mud, it got down to a certain layer, and all these rocks were just there. And all the real gooey mud ran off. All right, I am in basically the exact same area. I'm only a few miles away from this dinosaur state park in Connecticut. And they were making a new road or something, and they were cleaning it off, and they found all these dinosaur footprints, so they made a big state park of it there. And here it is right here. And I used to go there all the time because I serviced their business equipment. I used to go through this back door, sit around, have coffee with them and everything. When I found my stuff, I went there and it didn't work out well. Nobody likes this. They just wouldn't take it. They wouldn't even look. And I had a real argument with the last guy. He's still in charge there right now. It was only a couple of months ago. I said, this is just out of control crazy. You guys won't do your job. And he says, that's not my job. He said, I deal with dinosaur footprints. I said, well, I got human footprints. And I do. It's right here. It's a human footprint in the same rock they're talking about. And it's got the gray clay pushed down through the red bed. Watch this. Hold on. All right, I can explain this pretty well, I think. I am in a basin area, and what happened was after the Great Flood, all the boiling of all the body parts, the red flesh, the bloody red flesh turned into mud. And then the gray clay coated the next layer because it was a little later and it was mostly plant material and stuff like that. And then you had the black cap, and the black cap is here on top of this footprint, which the footprint pushed down through the red bed, which is nothing but mud. Most of my stuff is actual body parts, creatures. A foot. There it is in the picture right there. A foot. This is different. This was exactly at the time of this thing, the impact, and it had to be exactly. I mean exactly. This guy somehow survived the boiling and the first layer of flesh that eroded into mud. Somehow he survived. I don't know how, because right after that, the gray layer came on top, the red bed gray clay. Now this is gray clay. I have it right here. This is it. This is what you're looking at right there. There's a footprint. That's a footprint. Very, very easy to see. I've checked it out very closely, no question whatsoever. Plus it has what I call the suck-ups, which are right here. It had to be wet when it was happening. This whole thing had to happen exactly at the time this was happening. You see all those little dimply looking things? I call those suck-ups. And why those sucked up was it was wet and slimy, and he hit there running like hell probably. There's the balls of his feet. And went and pulled up these little things. Now on the edges where his foot hit, you see that there's no suck-up because there's no suction. There's the gray clay. This whole thing is gray. All the way down to the bottom, you see that? That's the gray. Somehow, everything here was wet exactly at the same time. And you can see way up here, you can see that this is pushed down. This gray is pushed down through that red bed. So he comes running. He hits this, it pushes this gray clay down through the red bed. So what we started off with was this. All right, forget the footprint for right now. But this had to happen between the red and the gray and the black. So the black is the fallout. And I don't think it took forever. There's no black on top of this here. All right, there's no black until you put this on top, and that is black as the ace of spades. So all of that was fallout from volcanism or whatever. So the underneath red bed is wet, totally wet. The gray is on top, totally wet, comes down through and makes a footprint. And Roger goes out and finds it. So this is, I don't see there's any possibility that you can dispute that. This happened at the same time. That was wet, this was wet, and then the black cap had to come afterwards. This was only, I don't know, a month, 2 months, a week. I don't have any idea. This was not 250 million years or 50 million years, whatever they say. All right, this, hopefully this clarifies as best I can. There's a substrate original layer that had to be here somewhere. Then we got hit by Venus. The whole Earth gets covered with this layer, and it is. The Triassic layer is worldwide. One gigantic ocean at that moment. It's sloshed in, sloshed out. Well, this is a sloshed into what they call a basin. So we're in this Connecticut Rift Valley basin. And I imagine the water's like up here somewhere. I don't know. Boiling all these creatures, and all the red came down here. And then the plant life and stuff, was just a little more buoyant, came down on top of that. And then the guy went running like hell through here. And then the black came after. It's the only possibility. We got a substrate. We had originally a full red layer. And we originally had a full gray layer. This guy came and he jumped running. He pushed the gray layer down through the red bed. And then the black came on top, and here it is. That's the gray pushed down through the red, and the black came on top. This couldn't have taken more than a couple of weeks. This is a tragedy that they will not allow this discussion. These are our educators. It's as bad as I can possibly even imagine. This has been a compulsion for me to straighten this thing out. This is just a mess. All right, you've seen mine, the gray clay, the red bed, the black clap, all that stuff. These are, they're saying, the oldest late Triassic footprint assemblage from North America. They're all over the place because the whole Earth, they're here. They originally found it in Germany. And I mean, it's just everywhere. And Yale went to Australia. I'm 15 minutes down the road, and I have a better selection of this stuff than anybody in the entire world. I'm not bragging, it's just true. All right, basically the only thing I didn't show you was the black cap. Now this was on top of here. It came in all three layers, boom. And it just separated all by itself. I didn't do anything to this. It just came up just the way it was. All right, the thing I didn't show you was this here is a black cap. All right, let me back this off a little bit so you can see. This is that in this way. So this is the top side of that, which is the black cap. And the black cap was right on here. That's this. When I found it, was all like this. This black cap was on top of the gray clay, which was pushed down through the red bed. It couldn't be any better than that. I mean, there's just no way you can get any better than that. And again, this is black. You see? That's the black cap. And that was right on top of here, which is this. So, all I can tell you is this is the Triassic signature. And that didn't take 50 million years to happen. That happened like in the blink of an eye. Okay, I have or will show you this this was sent to Yale March 4th, 2015. I had everything pretty much done. And I said it could have DNA in these samples, and it didn't work out well, as I'm sure I've already shown you or will. All right, I had so much evidence, I couldn't believe that nobody would talk to me. And this is the bloody end of a lung. This is the pleura that's on the lung. It's a very dense coating. It's a fabric. It's got to stretch and do this. This is an extreme fabric. You see it? I've shown videos of, under the microscope, my split leather cowhide belt. It's just actually the same. They got little tiny balls that are anchored all over the place in those straps, and they can only go to a certain length. And then, otherwise your lung would explode, I imagine. And this one here, this one's like brand new. I'm literally like brand new, and I drilled into here and got blood out of here, just as red blood as you can be. This one just bubbled blood out because the fascia had been already removed, and it took on silicates in this area. It was laying like this, and the blood ran down and ran out. There's a little flap on the bottom, just like this little flap here, and that was laying here, and that's where the blood ran out. Those are little blood blobs coming out of these little alveoli spots. See, they're all over the place. Now, again, I drilled into this and got raw red blood. I mean, and he said it was excellent quality. And I could show you, and not only that, a doctor, I believe it was a doctor, mentioned it. He says, oh yeah, that's the aortic arch, because the heart would be laying right here. The thing died flat like that. That is absolutely no question, because you can see the texture and the flatness of the back. You have to do some thinking. And this almost is transplantable. Now, you see that spot right there? It's just still got a whole ton of blood in it. And when you put a little water on it, it just turns bloody. And I'm not kidding you. I could take a scraper and scrape that right now, and blood would literally run out of there. And I was very careful when I did it. There was no contamination. I sent it off, and it tested out fabulously. All right, this was on the DNA test as well. I couldn't get any DNA because this was really turned into granite. This piece right here was still on, and I smacked it with a sledgehammer basically, and it popped right off like a scab. And I'll show it to you in a second. This is the fingernail. All right, it's a little slip of it has chiseled off basically, but this is how thick it would have been before, but you can still see it's quite obvious that's the fingernail. Now, vein and artery, this circular pad here, you see it? That circular pad is the little thing that goes in between bones so that they don't crush each other. Now, again, I had to break this off. This is literally from the back to the very tip of the front is somewhere around 36 inches. That's what I put down, 36 inches. So I had this tested. This is the 36 inch. I had this tested. This is the lung. And then I also had this hand tested. Hold on. All right, I had this hand tested, and I have fingers. Well, I have one good finger, and I have fingertips, several. And this is the pad. This is the left hand. Your thumb would go off this side. Small finger comes down this side. This is that pad. Right in the middle is the cleave. So you can do that. This right here is a tendon. If you layer it back, you're going to feel, you can feel that tendon right there in your own hand. And this is the pad that is your palm. Now, this silvery looking stuff is what I call it grip skin, and they call it traction, friction skin, whatever. It's the tough coating that's on your hands and your feet. And it's totally different than the rest of your flesh. It's different than your face. It's a different construction of keratin and kerogens and silicon, a lot of silicon. And it's just a tough, grippy stuff that you can do work with. And it'll wear off and there's no big deal. I'll tell you, the human body, what I have learned, it's just absolutely stunning. All right, so this is from the 36-inch fingertip. This is the piece I smacked off, and it just popped right off. My thumb is about the same width or thickness as a fingerprint. That's the sweat pores. You see them up here, sweat pores? And this peeled right off. It didn't fracture it's just like a scab came off. It's absolutely amazing. See, here it is right here. That was the whole fingertip I showed you before. And I smacked that piece off right up there, which is this piece. And it just peeled right off. And I can get right down into extremely excellent quality blood because it's just packed with blood. Fingers and toes are they're determinants of your blood. They're saturated. You see this? This is the area that I took the blood out of to send off to get analyzed. And they said it was excellent quality, it was dense, it was literally like red blood. This goes way back 10, 12 years ago. And it's just, when it was just fresh out of the ground, it was just, the blood was running off it. I'm telling you, that's just a fact. All right. Virtually all rocks are coated with what they call feldspar. This is my buddy here, Caesar the goose, Caesar Augustus. And these are his feathers. And the only reason feldspar is made out of this is because it's aluminum silicates. That was what was in the water. And it bonded with the collagens and kerogens. There's certain molecules that have certain affinities that bond with certain things, and the feldspar ends up being aluminum silicates, basically because phospholipids decay down to aluminum silicates. See phosphorus? That's what your membranes are made of, which is collagens and kerogens. So I tried to reconstruct. I did this maybe, 6 months or something. And it was coming along. This would have turned into feldspar eventually. I could see it was coming that way. I just, I don't know whatever happened to it, but this is what happens to the tendon. This is chicken. Now, there's some stuff I didn't understand when I did this. It should have had to be heated at a certain, I don't know how much, for how long. And there should have been transition metals put into here, which is the transition metals is from the blood. Because when you take chicken and you get raw chicken, they've already taken the blood out. So there was a couple things I didn't do, but I could see it was going to happen. Look, you can see it. Look at that. But I did put an electric discharge through this, a very slight electric charge, because that will. You have to understand telluric currents. And they just happen because light is hitting the earth, and as it passes, it charges. So I forget, a volt and a half or something. I had a copper wire here or there, I can't remember. This goes back a long time. But anyway, I knew I could make feldspar too. And now I can make it like that, no problem. All right, I had these tests done on 3 samples. I'm going to show you what the results were in a second. And I did the DNA extraction. Well, I didn't do the DNA extraction. I did the material extraction. Very carefully, very sterile, inside deep, no contamination, and it said there was no contamination. It was a very excellent quality, dense. Well, I'll read it to you. Anyway, this is back in 7/2015. Now, a year and a little more after that, a year and a half later, Yale basically said that they discovered all this stuff that I had. I presented all this stuff to Yale. That's why I'm upset. I mean, why shouldn't I be upset? Anyway, they did there's certain things they have to do, and he recorded every little bit in detail, and the guy was absolutely fabulous to work with. And he took a hit for this, for doing it. They did. I know, because he called me and he said, "Don't you tell anybody that I extracted that DNA or I extracted the sample." In other words, I didn't send him the rock and he put it in a clean room and did all that. No, absolutely not. I sent him glassine baggies, very carefully prepared, anyway. And here's the three I sent: 36-inch tip, the long, and the mud tip. Now, the mud tip didn't do so well. That was the one that really was turned into almost total mud. So, but they all had, and the negative controls were negative. See, negative control means is there more than one type of DNA in there? Now, this is the elution and shows the specific set of molecules in the DNA sequence that point to the fact that this was Homo sapien. Now, this is the words from him, and he says, "I stand by this." He said, but the only caveat is that he does not want to take claim for taking the sample and doing a clean room and all that stuff. No, absolutely not. I wanted to know if what I sent him, what it was. Now here it comes. The PCR products were submitted. So this is, he did all the stuff to create this sequence, to see the sequences that were in the DNA or in the sample. And they submitted it to Eaton Biosciences in New Jersey. DNA sequencing, the primer pairs that were used to generate the PCR products were also used in DNA sequencing. Excellent quality DNA sequence was obtained from the 36-inch tip, that's that monster, and the lung sample. They were just absolutely flawless. They were soaking wet blood. All right, and then and here's what it was: Homo sapien mitochondrial B loop, or cytochrome B gene, and a D loop region. Just can't get any more obvious than that. And here's all the sequencing. They did a lot of work, and it wasn't cheap. And I wanted it done so that there would be no questions to be asked, and there certainly was no questions asked. Tom Pachenko was the guy. What a very, very, very accomplished guy. And I know he took some heat for doing this. They don't like people that even think. It's just, I just can't even imagine anymore. I never expected what I ran into, and I wouldn't accept it. So I said, I have my own DNA test done. I did. I have my own CAT scans done. I did. I'll show you the CAT scans. There's a bone in the back. There's a tendon run up the side of your finger. And there's the other tendon on the other side. And the black and the white is the vein and the artery in the gray scale. And that's where the bone is. And that's a thumb. And you say, "Oh, Roger, how do you know that?" Well, because this side is flat and that side's bumped out. That's not your normal finger. That's a thumb. And it's actually a right-hand thumb. You can see a lot when you pay attention. And here's another shot of that. This is the 2D CAT scan. I also had the 3D done, but you can't see much inside and especially this, because this had really, really gone through the nucleophilic process. But there's still signatures. You see that? That's where the fingernail was. And this little hook right here is where the tendon locks in on the side of your finger, so you can do all that stuff. So, like I say, you pay attention, you can find things. And I found no appetite for that whatsoever. None. Zero. Just a fact. What we don't know about our past would just absolutely stagger your mind. And the only way we're going to find out about it is to sit down and think it over. You present your evidence, I present mine. I am not been allowed to present my evidence. This is it. This is all I can do is show it again, again, again. And that's all I can say, I suppose. Thank you. Love you all.